Back, and our fourth speaker is Beverly Rubin, who's the Associate Professor of Integrative Physiology and Pathobiology at Tufts Sackler School, graduate biomedical science. Boy, these names keep getting longer and longer and longer. So, yeah, boy. So let's add another two, right? Put a couple hyphens in there, right? In her research, focuses on the gonadotropin-releasing hormone, primary hypothalamic signal regulating pituitary gonadotropin secretion, and the essential function for reproductive fertility. And then our last and fifth speaker is Barbara Corky. She's been actively asking questions and participating, so you all know her, and she will be our last speaker. She's a Zoltan Cohen Professor of Medicine and Biochemistry at the Boston University School of Medicine, and serves as the director of the American Diabetes Association, Inc., and the Adipogenics, Inc., and as director of the citywide NIH-funded Boston Obesity Nutrition Research Center. And her main areas of research are obesity and the biochemistry of insulin in the adipose tissues. So we've got two final speakers that we've put to the end here so that they can really stimulate our discussion. So Beverly, take it away. There, they got you up there. That's the forward.
I'd like to thank the organizers for inviting me. This has been a very interesting day. I really look forward to the rest of today as well as tomorrow. And I'm going to be talking today about some of the work that we've done on the effects of perinatal exposure to BPA on obesity and metabolic disease later in life. And I'm going to give you basically no introduction, since we've had such a great introduction from Jerry and then from Bruce regarding obesogens. And so I'm going to start basically with bisphenol A. Oh, just so that we're all on the same page, bisphenol A is ubiquitous in our environment. 93% of the population has detectable levels of bisphenol A in their urine. We find bisphenol A in register receipts, in the resins lining tin cans, in dental materials, in plastic storage containers, and it is the building block of polycarbonate plastic. So bisphenol A is an endocrine disruptor with widespread exposure and multiple effects. And these are just some of the effects that we can attribute to BPA exposure. And the ones that we're going to be concentrating on today are primarily increased adiposity, glucose homeostasis, diabetes, and liver pathology. When we started the studies that I'm going to talk about, there was a controversy in the literature regarding early BPA exposure and body weight. There are many more papers now showing us weight gain in animals exposed to bisphenol A, but there still are several papers that have reported a weight loss or no change in body weight. Human studies have reported an association between BPA levels and increased body weight, waist circumference, and BMI. They have also shown obesity-associated metabolic changes, including insulin resistance and diabetes. So the questions that we were asking when we started these studies is, one, does developmental exposure to BPA alter body weight and body composition? And does it contribute to other associated components of metabolic disease? What BPA doses cause weight gain and increased fat mass? What is the critical window for BPA exposure? What about males versus females? And does early exposure interact with a Western diet in adulthood? So for the first part of the study, we did a dose response using 5 different doses, or 4 different doses and a control. We also looked at 2 different exposure windows. We had a perinatal exposure window that went from day 8 of gestation through lactational day 16, and we had a perinatal plus peripubertal time, which went from, it took the perinatal exposure and then added an additional exposure after the animals were weaned. We put them on drinking water containing the same levels of bisphenol A from postnatal day 20 to postnatal day 35. We looked at males and females, and we also looked at internal dose measurements to understand what kind of exposure these animals were actually getting. Here is our diagram of how we did our studies, and we exposed our animals via osmotic pumps on day 8 of gestation through postnatal day 16. When the animals were born, we culled the litters to 8, and then we weaned them on postnatal day 20 and examined that at various time points. That's the perinatal exposure. For the peripubertal and perinatal exposure, we just added this extra dose of bisphenol A, or extra time period of bisphenol A exposure, from postnatal day 20 to 35. And I just want to point out to you that the tolerable daily intake for bisphenol A is 50 micrograms per kilogram body weight, and the lowest observed adverse effect level is 50 milligrams per kilogram body weight. So our exposure levels, 3 of them were below the tolerable daily intake, and 1 was a little bit higher, but clearly well below the lowest observed adverse effect level. So here's our male data from the first part of our study, where we looked at body composition measurements, and you may recognize this from one of Jerry's slides, but these studies were done with the echo MRI, as Bruce is using now. And as you can see here, we looked at body composition in these animals, and as you can see, our controls are down here with percent fat, and our 25 microgram animals are showing us the highest percentage of fat, and the situation is reversed if we look at percent lean, as expected. The leanest animals are our 25 microgram animals, and the least lean are the 25 microgram animals, and the most lean animals are our controls. And if we use that data to calculate the fat to lean ratio, you can actually see, again, the 25 microgram animals have the highest fat to lean ratio, the controls have the lowest, and in between we've got our 2.5, here's our 250 microgram, and this is our lowest dose of bisphenol A here, the 0.25 microgram. In our P plus P females, our perinatal and peripubertally exposed females, we have a very clear suggestion of a non-monotonic dose response curve. Here we can see the fat mass in our animals, and you can see that starting at postnatal day 50, we see increases in the 2.5 animals that are significant. We also see increases not significant in our 25s, and that pattern continues throughout the experiment. So here's our controls in blue, and the highest dose, the 250 microgram in purple. And as you can see, if we look just at the 250 microgram animals, we wouldn't think there was any difference. And here's our body weight data. And here you can see, again, our control line is here in blue. And basically on top of it is our 0.25 and our 250 microgram animals. And then separating from the group are the 2.5s and the 25 microgram animals. And this is their body weights. So early into data collection for part 1, the perinatal and peripubertal exposure windows appeared to yield the same results. However, as the animals got older, it became apparent that that second peripubertal exposure exacerbated adverse effects in the females, but not in the males. Here we're looking at insulin tolerance tests for the P females, just perinatally exposed, and the P plus P females. And as you can see, these P plus P females show more insulin resistance than the controls. When we did serum assays, they suggested more pronounced metabolic issues with the P plus P females. Here we're looking at serum triglycerides, and as we can see, they are elevated in our P plus P females above where they are in the P females. And when we looked at liver extracts, at triglycerides in the liver of the P plus P females, again, we saw that the P plus P females were more affected than the P females. So is the peripubertal period, another critical window for effects of BPA exposure in females. Now, this is a very poorly studied window of exposure. We don't have very much data in the literature on this window, but in our studies, it appears that this may be a very important time of influence for BPA in females. And it's interesting that there is a recent paper analyzing methylation in 12-year-old girls, suggesting that BPA may affect human health through specific epigenomic modification in relevant pathways throughout pre-adolescent development. So that may be going on here, a definite effect of exposure in the peripubertal period. So if we summarize the data from the first part of the study, we see that there are differences in body weight and body composition influenced by BPA dose and by the sex. Although the P and the P plus P groups appeared similar at first, as the animals aged, body composition and metabolic parameters of the P plus P females were more affected. And internal BPA doses that were measured at the CDC in collaboration with Dr. Califat suggested that the doses of BPA were environmentally relevant. Here are the measurements of BPA in our animals, and as you can see, our control animals had no detectable levels of bisphenol A. Our 25 microgram females had just above the detectability of the assay, which is 0.3 nanograms of BPA per mil. And this is total BPA. They had no detectable levels of free BPA. And our 250 microgram animals had somewhat higher levels, as would be expected. But again, they had no mean free BPA levels that were detectable. So we're very convinced that these levels that we're working with are well within the range of human exposure. So in part 2, we decided to examine the effects of early BPA exposure combined with a high-fat diet, a 45% diet in adulthood. The doses were pared down for this part of the study. We had only the 2.5 and the 25 microgram BPA doses. We eliminated the low dose and the highest dose. The exposure window we chose was the perinatal window. It was most effective for the males, not probably the best choice for the females, but we had to make a decision. The diet was Harlan Tech Lab 2018 was our Chow diet, or the 45% high-fat diet from Research Diets. We looked at males and females. And the way we did this study is that we took litter mates and they were matched for body weight and body composition at 8 weeks of age. We had one male and one female from each litter placed on the high-fat diet, and one male and one female from that same litter placed on Chow for the remainder of the study. And this is what we saw. I don't know why this microphone is away. Okay, thank you. Oh, I thought, sorry, I thought I had to be next to one. Thank you. That's much better. Okay, so when we looked at body weight and fat mass over time in our perinatally exposed males, these are the Chow diet. Here are the body weights of the males that are control males. Here are our 25 microgram males in green and our 2.5 microgram males in red. Okay, so clearly the animals that were weighed the least were our control animals. The highest levels were in our 2.5, but they were very close and overlapping with the 25 microgram males. So there was clearly a difference in body weight over time, beginning at 9 weeks of age, because that's 1 week after they were put onto their diets. And then here, using the echo MRI again, we could see that the lowest fat mass was in our controls, and then our 2.5s and our 25s were higher in fat mass than the controls. Now this is Chow diet. When we put them on to the high-fat diet, we again saw that everybody got fatter, first of all. They all weighed more and they had more fat. But we see again that the controls stay lower than the treated animals. We were really disappointed, actually, when we saw this data because we thought that the high-fat diet would really make them fat. But then again, when you take a look at how fat these animals already are, I'm wondering if we've maxed out their capacity, because they're up there, and the mean weight here is 73 grams for a CD1 male. That's pretty big. And then here, fat mass, again, we see the controls are less fat than the treated animals. So again, I said we were really hoping to see a very dramatic change with BPA in terms of synergistically working along with the high-fat diet. We didn't see it in the males. Everybody increased significantly in body weight after 2 weeks on the diet. But we did see a difference in the females. In the 25 microgram females here, there does appear to be some synergy between the bisphenol A and the high-fat diet. Now, in looking at our animals, we did measure food intake every week, and we found not anything significantly different in how much food they actually ate. But what we did find that was different was the efficiency of their food. And here you can see how much food it takes, how many grams of food it takes to gain 1 gram of body weight. And you see that the controls need to eat a mean of 4 grams of food to gain 1 gram of body weight. But our bisphenol A animals, this 2.5 animals, the mean here is 3 grams of food to gain 1 gram of body weight. So it's a lot easier for these animals to gain weight than it is for the controls. All right, when we assess the metabolic parameters, here we're looking at evidence of altered glucose homeostasis in our Chow and high-fat diet males. And we can say that the high-fat diet does appear to exacerbate the effects of BPA on glucose homeostasis. So the disappointment in what we saw in just the body weight and body fat was sort of salvaged by the fact that other parameters were definitely affected by the high-fat diet in the BPA animals. So here we can see evidence of hyperinsulinemia occurring by 13 to 15 weeks in our males on Chow diet. Okay, so you see here that glucose levels were not any different. They were similar in all groups. But the amount of insulin it takes to maintain these levels is a lot higher, particularly in the 2.5 animals, than in the controls. By 22 weeks, again, glucose levels are very similar across all 3 groups. But in order to get these levels or maintain these levels, there's a lot more insulin that needs to be produced by the Chow males. Now here we have the high-fat diet animals. And as you can see, already their glucose levels are elevated, but they're similar across all 3 groups. However, insulin levels are very elevated in our high-fat diet animals at 13 to 15 weeks of age relative to controls. And by 22 weeks of age, we see that glucose levels are no longer being maintained. We now have severe hyperglycemia developing in our bisphenol A diet animals on high-fat diet. Okay, here's our insulin levels. They're pretty high all across the board. They're sort of maintaining things in the controls, but they are not maintaining glucose levels in the treated animals or the exposed animals. The hyperglycemia is strikingly bad. As you see here, if we just take the percentage of animals on the high-fat diet that had glucose levels above 350 milligrams per deciliter, you can see that approximately 37% of the 2.5 animals and 50% of the 25 microgram animals had levels of glucose circulating that were greater than 350 milligrams per deciliter, and that's very, very high. And many of these animals became diabetic. They just went off the scales of the meter a week or 2 after this. Right, we also saw evidence of increased inflammation in the adipose tissue of our BPA-exposed mice, and it was exacerbated by the high-fat diet. Here you can see the crown-like structures in adipose tissues, which are arrangements of macrophages around the remnant triglyceride of dead adipocytes. And this is often associated with adipose tissue inflammation and systemic insulin resistance. These are the MAC2 staining in the gonadal fat of the mice on a Chow diet. Here you can see the controls, and here you can see the 25 microgram animals, and you can see this rim around lots of the adipocytes, which are dying. And then you can also see the normal, or somewhat normal-looking adipocytes, but they're a lot larger than the ones in the control animals. So this really looks like not such great adipose tissue, and this looks fine. So the males are being affected by the BPA, the early BPA exposure. When you look at the number of crown-like structures in the Chow animals, they're significantly higher in both groups of BPA animals relative to controls, and that's true in the high-fat diet as well, but there are more crown-like structures seen in these animals than in the Chow animals. And as we would expect, macrophage and inflammatory cytokine expression was increased in the adipose tissue of these animals. This is the high-fat diet animals now. What about liver? Now we know that liver is very important in controlling glucose homeostasis. So when we looked at liver, we saw increases in cholesterol in the bisphenol A exposed animals, increases in non-esterified fatty acids, and some increase in triglycerides, although it wasn't statistically significant. When we looked at lipogenic and adipogenic genes in the livers of these animals, we saw significant increases in many of the lipogenic and adipogenic genes, and we also saw very significant increases in the cholesterol synthesizing genes. So in summary, early BPA exposure increased body weight and fat mass. It altered body composition. It altered parameters of glucose homeostasis, including hyperinsulinemia, insulin resistance. High-fat diet exacerbated effects led to severe hyperglycemia, eventually culminating in a loss of beta cell mass. We saw increased evidence of adipose tissue inflammation, and that effect was exacerbated by the high-fat diet. We saw altered liver parameters. We saw increased fat accumulation, steatosis. We saw increased expression of adipogenic and lipogenic genes. The high-fat diet animals also showed an elevation in inflammatory gene expression. So the effects of BPA on body weight, body composition, and elements of metabolic disease differ with BPA dose, sex, exposure window, and diet. Now, since the start of this work, there's been a growing body of data from studies in mice and rats to corroborate our findings that early BPA exposure may act as an obesogen. It may alter body composition, glucose homeostasis, and may affect liver function. But there are a lot of important questions that remain. Probably the most important is, what about humans? Now, I told you before that epidemiological studies reveal associations of BPA levels with various parameters of metabolic disease in adults. We also know from the work of Tristan that it also is shown in children, that children and teens also show a correlation between bisphenol A levels and body weight. Now, I was going to say that to date there are no reports of a correlation between prenatal and neonatal BPA levels and increased body weights in children, but I think that that's probably not true, because Danya did publish something in which she showed a correlation between maternal bisphenol A levels and children's BMI at 40 years of age. There's also a recent report of an association of early BPA exposure related to increased leptin levels in 9-year-old boys, and this was from Kim Harley's group in California. I think that it's just too early to know about the story of prenatal or perinatal exposure in humans, because the studies are being done now. It's going to take a while for us to see this, particularly if really the time that changes are occurring is in adulthood. We have a long way to wait to see the results of some of those studies. And most importantly, what are the mechanisms through which BPA is working to affect these endpoints? Well, we know BPA has estrogenic actions, and DES and genistein can increase body weight when given perinatally. So that's in that category. We know that BPA can affect directly adipose tissue, pancreas, and liver. We know that BPA has effects on other endocrine components, including thyroid and adrenals, and these, of course, are very important in weight regulation. Developing circuits for food intake and metabolism, that's going on during the period of exposure in our animals. So we really want to take a look at this, not so much for food intake, but for metabolism. We know that there are epigenetic changes. We've seen them in our own animals in liver with the collaboration with Angela Slitt at URI. We also want to know about the microbiome in these animals, because clearly these animals are being exposed at a time that's very important for microbiome development, during gestation, during birth, and during lactation. We have a little bit of very interesting preliminary data that suggests that good bacteria are decreased in our females at 5 months of age, and the bacteria that signify or that are seen in intestinal pathologies are increased in our animals. But that's just a pilot study, and we'd really like to explore that finding further. And then the fact that there are altered metabolic pathways. Now, we've just started to do this work. We've been looking, along with, in collaboration with Danielle Zalco in France, we've been looking at metabolic fingerprinting in our animals. And clearly, we see different metabolites. And eventually, we hope to figure out what kinds of changes in metabolic pathways are occurring that will be influencing the body weight story. So I'm not going to have a chance to show you that data right now, but I would just like to acknowledge my collaborators, Andy Greenberg, Marty Obin from the HNRC at Tufts, and Anna Soto, who's been a longtime collaborator. And we have several other people working on components of this, including Angela Slitt, Tom Jaton, Antonia Califat did the measurements for us, and Daniel Zalco and Nicholas Cabaton are working on metabolic fingerprinting. And then in the lab, we just have a crew of people who have spent a lot of time dealing with a lot of animals. And I thank you for your attention.
Any follow-on questions of clarifying points? Yes.
Hi, Melissa Perry, George Washington University. That was an elegant set of studies. Thanks for the presentation. So you're working with a difficult contaminant, and that's BPA, because it's fast-acting, non-persistent, and fleeting. And so I was intrigued to understand your dosing scheme and what insights you can give us as to the difference between the 2.5 and the 25 micrograms, and if you were able to assure steady state. So I'm sure you've been working hard on that. And then secondly, I noticed that in at least a couple of instances, you had the prototypical nonlinear or non-monotonic dose response that we see with a number of other endocrine disruptors. Mm-hmm. And so can you give us any insight into surprises that you saw with respect to increasing dose?
Well, I guess we thought the 250 microgram dose would give us better effects than our 25 microgram dose, but that wasn't the case at all. If we had just looked at controls in 250s, we would say there's no difference in body weight. And it's very interesting because a paper that came out in endocrinology a little while ago from Wei and his colleagues in China, it was very dramatic. They had this beautiful paper showing differences in rats with 50 micrograms of bisphenol A. But when they gave their animals 250 micrograms or 1,250 micrograms, they saw nothing. They had no effect whatsoever. So it is very clear that the low doses of bisphenol A are the effective ones here. And once you get across a certain border, it no longer has the effect, which may explain why there's a controversy in the literature. I think it really depends on strain, dose, time of exposure, and lots of different factors that can all enter into this picture. So we've just tried to keep things as stable as we can, and the way that we give our bisphenol A, we started out giving it in the water, but we were very concerned about that because we couldn't really tell if every animal had the exact same amount of bisphenol A, because you know that the mice like to play with the water. So we would measure water every day to see how much they drank, but we weren't really confident that we could really tell how much each animal had gotten. So we went to osmotic mini pumps, and we did lots of assessments to try to see if they actually released at the same rate that they said they did, et cetera. We did all kinds of tests to find that. And then that's what we used to expose our animals. And at the end of the period, we would take the pumps out and just make sure that they had expended everything inside of them, so that we knew the pumps were there, they should have been operating, and they did release their contents. That's how we did it.
Hi, Liza Mikowski from UNC Chapel Hill. I was really intrigued by your macrophage findings and the adipose tissue and the inflammatory effects. Are there known effects of BPA on macrophages themselves, or do you think this is an indirect effect through adipocytes?
I don't know if there's an effect on macrophages themselves. I think that the tissue, the adipose tissue, is no longer healthy tissue. I think the bisphenol A is really making the adipose tissue dysfunctional. I mean, these tissues, if you look at them early on, you see that the cells are much larger in the bisphenol A exposed animals. So they're packing in a lot of lipids, and I think eventually they just give out. And what we're looking at is the macrophages coming in to clean up the mess. But it's definitely dysfunctional looking adipose tissue.
Could you tell us what the hormone levels are in these animals when you sacked them? In other words, what are the estrogen, androgen levels, the thyroid hormone levels?
I wish we had that data. Unfortunately, we don't. We have done estrogen levels on our animals during a normal cycle. We've looked at estrogen levels on the day of proestrus, and we did not see any significant differences between the treated animals and the control animals, at least with our females. That's the ones we were looking at. I don't have these hormone levels, unfortunately, because at the end of the study, we did take the serum for all kinds of things to look for cytokines, adipokines, and various other things. And as you know, mice are pretty small, so it's pretty hard to get all the measurements you would like. We have measurements of leptin, they are significantly increased. We have measurements of adiponectin, they're significantly decreased. So we have a lot of measurements in our blood, but we do not have measurements of thyroid hormone or corticosteroids. Although, no, that's not true. We do have corticosteroid measurements, and they seem kind of normal, at least.
So Joseph Kroll has published studies suggesting that BPA is actually hypothyroidic. Mm-hmm. And so the question would be is whether this effect is through a hypothyroid type activity or whether it's really an estrogenic activity.
Well, I think definitely that, I mean, there could be some estrogenic component, but I am certain that there are components of other factors of the endocrine system, particularly thyroid hormone. And there is evidence that the stress axis is disturbed in bisphenol A treated animals. We haven't checked it in our animals, but that could be another contributor. Thank you for that comment. That's a good comment.
Okay, thank you.